So, in last class, we talked about this classification. Now, do you remember this classification? 6 type of enzymes.
To be honest, not really. Can you review them?
Hello, can you hear me?
Yeah.
So there are 6 types of enzymes, and you have to memorize according to number. 1 is always oxidative reductase, 2 is always transferase, 3 is always hydrolase, 4 is always lyases. Lyases breaks bond, and 5 is for isomerase, 6 is for ligases. Oxidative reductase, we will talk about each of these. Oxidative reductase does oxidation reduction. So these are the classification based on function. Actually, I have told you specificity. We are talking about a specificity of property of the enzyme. So what does specificity means? A particular enzyme will catalyze a particular reaction. So we have seen how many properties? S3 and R3. I told you. S3 means speed, selectivity, and specificity. R is reaction site or active site, and then reaction condition, and then regulation. So 6 properties we will be talking. Whatever whole enzymes topic, we will talk about only about properties. So see, S3 in S3 as specificity. Specificity means specific type of reaction the enzyme catalyze. So enzyme is divided based on the reaction the enzyme catalyze. So you can see we group enzyme into 6 categories. First category is oxidative reductase, performs oxidation reduction. Second is transferase, it transfers. Third is hydrolase, it hydrolyzes. Fourth is lysis, it breaks the bond. And fifth is isomerase, and sixth is lysis. You cannot put oxidative reductase here. So you have to remember, I have given you the formula also, OITH, OTH, LIL. And I have given you tricks to remember them also. So let's start with the. So I have already explained what is EC number. So this is class, this is subclass, this is subclass, and this is, sorry, this is class, this is subclass. Yes, this is subclass. You can see subclass, and then this is serial number. Serial number given to a particular enzyme. So you can see how what happens is we will, let's draw a box like this. So this is the enzyme, the container containing all the enzymes. Just remember like that. So let's divide into a 6 group. So let's divide into a 6 group. 1, 2, 3, 4, 5, and 6. 1, 2, 3, 4, 5, 6. So when you give number 1, so enzyme will come into this box. And then again, next, if you put the next number, so enzyme will come into sub box. And then again, if you put some number, it again come under box under box. So it's sub box. And then if you give the last number, it will specify a specific number. So that is how you can remember this enzyme classification this pictorially. So actually we are very specifying enzyme according to its function this way. Now, so this is the enzyme of creatinine kinase. It is a transferase, that is 2 is transferase. So we are describing this enzyme. So for your concern, just focus on this first number on class. Even if you don't actually memorize, you don't need to memorize. You must not memorize this, or just understand these numbers. That's it. But you have to memorize the first examples of. So I have given you this all points. So oxidative reductase always utilize coenzymes. So NAD plus and NAD plus. Most transferases also utilize enzymes. See, oxidative reductase always utilize NAD plus. Generally, you will see NAD, NADP, FAD, FMN. So this cofactor oxidative reductase will utilize. Remember this. And then most of the transferase will also utilize what? Coenzymes. Ligases always utilize ATP. That is a very important point. Lyases never utilize ATP. The difference between ligases and lyase. Hydrolase always utilize water to break the bond. Isomerases bring about intramolecular changes. So this is the very important points that I have given here on one slide. Now let's talk about the oxidoreductase. Oxidoreductase, what does oxidoreductase do? Oxidoreductase brings about, it does oxidation reduction reaction. So it, whatever enzyme that is involved in oxidation reduction reaction, those are oxidoreductases. So how do we name this? How do we give systematic name? We give this systematic name through this. So donor, acceptor, oxidoreductase. There is a number given, and the second method is you give this type of name. You write donor name and then this semicolon, acceptor name, and then you write oxidoreductase name. Donor, acceptors, oxidoreductase. And then based on this only, the name is also given, number is also given. I will explain that. You can see here, this oxidoreductase, the common name, we will use 4 common name of this oxidoreductase during our, when we talk about pathways. These are, first common name we will use is dehydrogenases. So see, examples are very important. And second is oxidase, third is hydrolase, first is reductase. So dehydrogenase, oxidase, hydrolase, hydroxylase, and then reductase. So this is the very important example of hydroxylase, cytochrome 450 enzyme. So remember, this is a very important example of hydroxylase. I am pointing on this only. So remember at least this only. Other you will be remember, because dehydrogenases will be coming many times, and in enzyme catalyzing dehydrogenase reaction, you will see the dehydrogenase here. An enzyme catalyzing reaction oxidase, you will see oxidase writing here. Enzyme catalyzing reductase reaction, you will see reductase. But enzyme catalyzing this hydroxylase reaction, you will not see hydroxylase. You will see, but you will generally, it will come as this cytochrome 450 enzyme. So you will have to remember this cytochrome 450. Even hydroxylase is not written, so you will have to understand this is cytochrome 450 system. So remember, this is present in endoplasmic reticulum, mitochondria, and mainly membrane bound. These are membrane bound enzymes. So we will talk also later also in detail about this. This is a very important enzyme, cytochrome 450 system of oxidoreductase. Now see this. Let's take the example of this 7-dehydrocholesterol reductase. So this enzyme is the enzyme, last enzyme of cholesterol synthesis. Now reductase, as the name suggests, reductase, it is an oxidoreductase enzyme. So what the first, so because it is oxidoreductase, you give like enzyme commission number 1 first, and then what rest of the number follow based on what is the donor here and what is the acceptor. You can see the donor here is, the electron donor is CH group. That is what this represents 3. So if you go into detail, you will remember. You don't have to go into detail. That is why just remember this is oxidoreductase, and this number actually defines this oxidoreductase reaction more specifically. Okay, this represents one. The first one represents it is oxidoreductase enzyme, and then other number defines that reaction more specifically. That is what you have to understand. Okay, and you don't have to memorize, just understand this. So this is oxidoreductase. This is 13121. This is last is serial number. This is class, subclass, subclass, and then serial number. So as I have told you, you make a box. So this is class, box is class. This is subclass, and then again you make a subclass, and then serial number. So this is how enzyme is specified. Every enzyme is specified by giving a number based on the type of reaction that enzyme catalyzes. Now let's talk about other examples. So lactate dehydrogenase. So you have, these examples are very important. Okay, all examples are very important. You can see reductase. From that you can easily understand this is oxidoreductase. Here you can see dehydrogenase, lactate dehydrogenase. So it is extracting hydrogen. That means it is extracting hydrogen from this molecule. You can see from this molecule, this is a alcohol is a donor here. So donor and acceptor is NADH here. NAD is acceptor. So from this NAD extract the proton. So number 1 defines this is a oxidoreductase, and this defines which this one actually represent what is donor, alcohol, and this one represent this donor. So the acceptor, donor, acceptor. You don't have to go into detail of that. Just remember one is oxidoreductase, and this 4 number is given to specifically actually specify that enzyme, the type of reaction. If you go into detail of this classification and memorize those all things, you will easily understand based on this number, okay, this particular enzyme does this thing. So you will this is the power of this classification. Okay, so you don't need to go into subclass, subclass. Just understand this. Only you need to memorize this class. This is oxidoreductase. Second enzyme is glucose 6-phosphate dehydrogenase. This is also because it is dehydrogenase, it is also placed into group of oxidoreductase. Now let's talk about transferase. So what does this transferase do? Transferases transfer one molecule from other molecule. As name suggests, it transfers. So this reaction, what is the reaction type? It transfers. So you can see this example here. You can see this is glutamic acid, an amino acid. From this glutamic acid, what happens? You can see change between this glutamic acid. Here you can see amino group. Here you don't see amino acid. In place of amino acid, amino group has been replaced by this oxo group. And amino group is been transferred here. So you can see amino group here. So this is actually transfer of amino group from this compound to here. So this is called transferase. This is a transferase of amino group, and that is why this is given number 2. And then based on the particular reaction that it catalyzed, then it will be given next number, next 3 number. So you don't have to worry about that. So just understand the next 3 number is actually defining this more specifically, more precisely. That is what you need to understand. So this is called alanine transaminase. This is a very important enzyme that you will be doing in laboratory test, that you will be using in laboratory test also. So you will be doing this test when you become a doctor. So alanine transaminase. This is a very important enzyme. You will be doing this test when you become a doctor to predict the liver, the functionality of liver. Alanine transaminase does this reaction. So again, systemic name is donor acceptor grouped. So name of group and then amino transferase. So in many cases, donor is a cofactor, as I've told you in the before only. As I've told you before only, you can see here. So most of the transferases utilize coenzyme, and all oxidoreductase utilize, but most of them. That is what the same thing is repeating here in a different way. So you can see, so donor is cofactor. Generally, donor is a cofactor, charged. So donor will be charged on the cofactor. Even if you don't understand here, what happens? I will here for, let's take the example. There will be an intermediate. Vitamin B6 will be here for this enzyme. And then what happens is this amino group will be taken by this vitamin B6, and vitamin B6 will add here. So intermediate, that is what is. So donor actually will be a cofactor. This cofactor will carry this group and then transfer. This donor will be an intermediate, an agent, that is cofactor. So that is what this statement says. Now let's see other example. The same way ALT, you see AST, spartate amino transferase. Let's see the example of this spartate amino transferase. This is donor. So donor, and this is acceptor. So donor acceptor group transferase, group then transferase. And number is given based on this. This 2 represents the class to which this enzyme belongs. This belongs to transferase, 2 for transferase, 2 for 2. And then 6 represents the type of group you being transferred. So what is the type of group? Type of group is here amino group is being transferred. So this amino group is given 6. And then more specifically, this group is specified more specifically by this next number. You don't have to memorize this. Just understand this is 2 group fall into 2 group, and it transfers amino group from this to here. Here it transfers here. You can see here. So there is no amino group, and there is amino group here at this position. And this is mediated by vitamin B6, pyridoxal phosphate. Pyridoxal phosphate. So you have to remember, in previous class I have given the slide. So you have to remember what group transfer what. Pyridoxal phosphate transfers amino group. So these are the questions. Pyridoxal phosphate. Now alanine amino transferase, I've already given example in previous slide. Here this is alanine amino transferase, ALT. And then creatine kinase, you can see this creatine kinase here. This is creatine kinase. In the first example is creatine kinase. This is creatine kinase. I've given in the beginning only. This is creatine kinase. So you can see second group 7. Then based on type of, because it transfer phosphate group, that is why it is given number 7. So pyruvate kinase. This is pyruvate kinase. You can see this is pyruvate kinase. So pyruvate kinase transfers again phosphate group, and that is why it is given number 7. So it represents the type of group. Second number represents the type of group transfer. First number represents the function, what it does, and then second number represents type of group. So this 7 represents, actually 7 number represents phosphate group. And let's see this example very precisely. So all kinases are transferase. You know, all kinases are transferase. You have to remember all kinases are transferase, and you will talk about these kinases. So have you gone through this cyclin dependent kinase? Anybody please answer me quick. Cyclin dependent kinase. Be quick. No?
No, we haven't.
So cell cycle, haven't you been taught cell cycle?
Like mitosis?
Yes, mitosis, after mitosis, cell cycle.
We went over meiosis a lot. We have not gone over mitosis yet, though.
No, meiosis, mitosis, anyway, the cell cycle. G phase.
Oh, yeah.
Have you gone through?
Yep.
So this cell cycle, in cell cycle only, this cell cycle transition happens because of cyclic dependence. So you must have been taught, you have forgotten. You must have been taught. Yeah, so this is cyclin dependent kinase. Yes? S phase, then S phase 2, G, this is G1 phase, from G0 phase 2, this is G2 phase, then M. So this is what happens. This is cell cycle, and each cell cycle requires a specific type of cyclin expression. You see, cyclin D will be expressed first. Then cyclin EA will be here, EA. And then here one cyclin B will be here. So like this, cyclin expression. This is called cyclin dependent kinase will be here. So just remember, for kinases, just forget about this right now. Any kinases, what does, you remember this picture. So kinase, what does kinase do? Kinase is an agent. Just remember this enzyme is an agent. It actually take the phosphate. This is ATP molecule. Adenosine, this is adenine, this is sugar, pentose sugar, and this is triphosphate. So this kinase takes phosphate group from ATP and this transfer phosphate group to a substrate. Substrate can be glucose or other substrate. A substrate can be protein. Did you understand this picture? Tell me.
Yes, doctor.
Yes. So what does kinase do? Kinase is an enzyme. It is a very important enzyme. See, you will be everywhere you will find kinase coming repeatedly. So if you understand this principle here, so my purpose is to provide you with principles, facts that you can utilize every time, everywhere. So I am providing you like a sort of like route to the knowledge of the medical knowledge. So this is like route. So if you understand this much knowledge, you will understand all kinase in very deep way. So you will find kinases coming to physiology, kinases will come in biochemistry repeatedly, kinases will come in cell cycle repeatedly, kinase will come in your pathology repeatedly. So kinase, you will see everywhere kinase is coming. So if you understand this simple fact, it will require maybe 5 minutes to memorize you, but it will have a huge impact if you understand in a very deep way. So understand this. So what does this kinase do? Kinase pick this phosphate from this and transfer this phosphate to this. After picking, so what will happen? The ATP will convert into ADP. So you can see what does kinase do? Kinase pick this phosphate from ATP and then transfer to substrates. So this is how this does reaction. So you can see this is substrate and this is ATP. So this is kinase. So what does kinase do? Kinase transfer a phosphate ATP. ATP means phosphate will be 3 phosphate will be there. So it will transfer a phosphate from this to this, and then substrate will be phosphorylated, and then ATP will convert into ADP. So the enzyme that does this function, that transfer phosphate from ATP to a substrate, is called kinase. Remember, kinase means always it deals with ATP. So kinase. So kinase can be glucose kinase if glucose is added when ATP, when phosphate from ATP will be added to this glucose 6-phosphate will form, and then ADP. This is called glucokinase. When this
Dr. Santosh.
Yes, tell me.
Yeah, there are students waiting in the waiting room. They are being kicked out.
See, every time I cannot do in between.
No. They had some internet issue. They just got kicked out.
Okay.
They were before there, but now they got kicked.
Okay, no. So this is what happens online. This is one also a problem of online teaching. Anyway, no problem. So you can tell me orally, because what happens is I don't see any while teaching on this software, this Windows 365. I don't see anything, message or anything. I don't see anything. Anyway, so now, so if the substrate is protein, and if protein is phosphorylated, it is called protein kinase. If protein is phosphorylated, it is called protein kinase. So these are the protein kinase: cyclin dependent kinase, protein kinase A, protein kinase C, myosin light chain kinase. These are the important kinase you will be encountering when you study after study further in next units, in next modules. So substrate kinases are hexokinase, glucokinase, phosphofructokinase, and pyruvate kinase. And this is kinase is very important transferase. So kinase is a transferase. Remember, kinase is a transferase. It transfers phosphate group from ATP to substrate. So now next very important enzyme is phosphorylase. Okay, the phosphorylase. What does phosphorylase do? Phosphorylase use phosphate to cut the bond between two carbohydrates. Say glucose, between two glucose molecules, it use phosphate group to cut. So this is called phosphorylase. Remember the name of the enzyme. This is also placed into the group 2. So phosphorylase is also a group 2 enzyme. You can see this is phosphorylase. This bond between two glucose, and glucose will be, the bond between two glucose will be cut by this phosphate group. So phosphate will be added here in one. So this is called phosphorylases, and the enzyme that does this phosphorylasis is called phosphorylase. In that sense, it is transferring phosphate group to glucose. You can see phosphate group is being transferred to one of the glucose. That is why it is placed into transferase. So glycogen phosphorylase is a very important enzyme, a rate limiting enzyme of glycogen breakdown. So remember these two very important example. One is kinase. You can see this is also pyruvate kinase. Here you can also see kinase. So by now you know what is the function of kinase. You know what is the function of phosphorylase. Now let's talk about this hydrolase. So hydrolase, if you saw hydrolase, what does it mean? It is very easy. It always utilizes water to break the bond. Water to break the bond. That is what hydrolase is. All digestive enzymes are hydrolases. All digestive enzymes are hydrolases. You can see here, hydrolases are this digestive enzyme, amylase. So this alpha amylase actually breaks the bond of carbohydrate. then trypsin is protein. The protein of amide bond of like peptide bond of protein will be digested again by utilizing water. So this all enzymes are hydrolysis. Chymotrypsin is digestive enzymes of intestine secreted from pancreas. It is also digestive enzyme for protein. It is also digestive enzyme. It is digestive enzyme for carbohydrate. Now acid phosphatase and alkaline phosphatase have different places. So alkaline phosphatase is present in bone. It releases it breaks the phosphate bond. When phosphate, it actually breaks the phosphate bond. That is why it is called phosphatase. Acid phosphatase is present in prostate, and it also does the same thing. But this is alkaline because it acts in alkaline environment. It acts at pH 10. It is acidic because it acts in acidic environment about pH 3. So that is why this is acidic phosphatase and alkaline phosphatase. Now let's see. Now you can see this enzyme actually class is numbered. First number will be 3, but next number will be the basis of next number will come from which type of bond this hydrolase break. Whether it break the carbon oxygen bond, carbon nitrogen. So the type of bond next number will define the type of bond. So let's see the example. First example you can see here. This is acetylcholinesterase. So this is called acetylcholinesterase, and other name, you can see the system, a bit systematic name, acetylcholine acetylhydrolase. This is a hydrolase. So this hydrolase breaks the bond here, this bond, and when it breaks this bond, so this is given, because this is a hydrolase, this is given number 3, third group, and this defines the type of bond breaking. So this is a ester bond, and this one represents ester bond. So you don't have to go into detail, just remember I have given you a glimpse of that. So this is ester, this one represents ester bond. This carboxy ester bond, one represents carboxy ester bond. So when you go through this enzyme commission number, EC number, so you will definitely find, okay, what type of reaction is being going on from this. So type of reaction. Yes, any problem?
So if it's a 3, how you figure it out with the number given, with what kind of reaction is this?
Yes. Because I know whole classification. And as a matter of fact, you don't have to go into detail of the classification. You need to know only 3. If you can classify this is as transferase, that is where this is hydrolase, that is fine. Okay. Okay. If you can recognize, okay, this enzyme is hydrolase, so this is more than enough. But I can understand this enzyme from this number. Okay.
Yeah, got you. Thank you.
Okay. So if you want, then I can help you, but you don't need to understand that. It will never be true. Just if you memorize this number 3, okay, this present into hydrolase, this utilize water to break the bond, that's enough. Okay. Now, so this is also, you can see this is alkaline ALP means alkaline phosphatase. This is a very important, this is again a very important enzyme that you will be using in liver function test, doing liver function. So enzyme has wide implication for understanding your metabolism. To understand, if you understand enzyme, you will understand metabolism. You will actually, some enzymes are also utilized as drug. You will understand the pharmacology, some part of pharmacology better. You will understand some enzymes, many of the enzymes are utilized as laboratory test. You will understand laboratory test better. So enzyme is very important. That is a very, very important topic, a very core of biochemistry. And that is why for the timing, it may feel like boring, but go through this. Use your time. Once you go through this boring kinds of thing, you will find biochemistry very interesting. Okay, so this is alkaline phosphatase, very important enzyme. This actually breaks the bond utilizing water, phosphate bond from organic compound, and acts in alkaline condition present in bone. And this is the enzyme that is required for bone formation. Okay, alkaline phosphatase is required for bone, for mineralization of bone. Now alkaline phosphatase breaks this bond, and you can utilize water, and that is why this is placed into group 3. And this next number is given to the type of bond it breaks. Just remember that type of bond, they represents type of bond. Now liases. So liases. Yes,
So I have a quick question. So let's say for amylase, so it comes from the pancreas, right? So all it does is it uses water to, like.
It does not come from pancreas.
Okay.
This enzyme come from pancreas. Okay, these three.
Oh, okay.
These are digestive enzymes.
Right.
Okay. Then, yes, tell me.
Thanks.
Okay. These are not these are the bone. This is present in bone. This ALP and this is same. This is
Dr.
ALP.
Francis. Someone else got
Okay.
kicked out.
This hydrolase. Let's talk about lyase. So lyase, you can see lyase is means breaking the bond. So it breaks the bond without utilizing water. So here you have seen utilization of water. Okay, utilization of water. Here you see without utilization of water, especially without utilization ATP and water. You can see here this is an enzyme, aldolase enzyme. This is a part of glycolysis. So this is, you can see 6 carbon is here. 1, 2, 3, 4, 5, 6 carbon. With both in there are phosphate. This is called fructose 1,6-bisphosphate. So this is a carbohydrate. Glucose and fructose are similar type of carbohydrate monosaccharide. So this will be broken down into 2 groups here. So you can see broken down here. It will form 3 carbon this and this 3 carbon this group. So this is dihydroxyacetone phosphate. We will talk about this in detail during glycolysis. Don't worry about this. So this is glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate. Now this breaking down, can you see anywhere utilization of ATP or anything? Water? No, it does not utilize ATP or water. And this reaction is reversible reaction. Okay? So if you see opposite reaction, if you see this reaction, you will see the breaking of bond. But if you see opposite reaction, you will see, okay, it is joining of bond. And then you will confuse that it may be a ligase. But you know, ligase always utilize ATP. So this is the difference. But it does not utilize ATP. I'll repeat. If you see the opposite reaction, you will see the 2 compounds being joining. Joining means ligase, but it is not ligase because it is not utilizing ATP. So in order to call any enzyme ligase, that enzyme must utilize ATP. Did you get that?
So lysis is basically a reversible or irreversible.
Yeah, reversible. Lyases can be reversible, but ligases are irreversible.
Okay, got you.
Okay, so now let's see this. So you will see here, I told you, this is one confusion. People get confused between always get confused between sometimes ligase and versus lyase. This always utilize ATP. Always utilize. This never utilize ATP. This is a very big difference between lyase and ligase. Now, you can see other example I'll give you. Other confusion arises. So you can see there is a utilization of water. But if you carefully see, this water is being utilized to break the double bond. While in hydrolase, water is used to break single bond. Got that? Hydrolase, so I am adding one more thing. Hydrolase breaks single bond utilizing water. Okay? There is something called hydrolysis. Now hydrolysis will break this double bond, not the single bond. It will break the double bond. If some enzyme breaks the double bond utilizing water, it is called lyases. Which lyases? Hydrolysis. So don't become confused between, here you don't become confused between lyase and hydrolyase. So fifth enzyme is isomerase. You can see isomerase, many types of isomerization happens. So these are the type of isomerism. You must have studied in organic chemistry. Racemases, epimerases, cis trans isomerases, other isomerases, tautomerases, mutases, and cycloisomerases. So these are the type of isomerases. And see, the first number given to this number 5/5 is given to isomerase, and the next number actually give clue about this type of isomerase. Okay. Next number will give the clue of type of isomerase. Let's take the example. So this is an enzyme of glycolysis again. So you can see this glucose 6-phosphate is converting into fructose 6-phosphate. This is called phosphohexoisomerase. This is phosphohexose isomerase transform this glucose to fructose. So when it isomerizes glucose to fructose, because both are sugar, one is aldose sugar, other is ketose sugar. And isomerase name given to 5. So 5 number is given, and this 3-1 is given to the type of isomerism this enzyme brings about. So you don't, I told you, you don't have to actually memorize, just understand this last number, further number actually explains this first number more precise. So this enzyme is 5319. Now ligases, let's talk about ligases. So ligases, I have ligases means they join the bond, two bonds. They join the two.
Can you repeat
Yes?
the last one, the previous one? Can you repeat it, please?
Yes. So this is isomerase. Okay. Can you see? This is isomerase, phosphohexoside. That is why it is given number 5. And this next number represents type of isomerism, isomerization. This type of isomerization. This enzyme does. Got that?
Yeah, thank you. Thank you.
Okay. So this is like a bit systematic written name. You can see D-glucose 6-phosphate, aldose, ketose, isomerase. This is also systematic name that is given into word. This is not recommended. We will not be using this name during reaction. But I will tell you this what type of classification. When I teach you this pathway, I will tell you which class does this enzyme belong. I will tell you the class.
Yeah, but we don't worry about 9, right? And 1, as you said before. We don't.
You don't. Yeah, you don't worry about this next numbers. Just
Yeah.
worry about this only. So if you are able to classify enzyme, that is more than sufficient for your understanding.
Thank you.
Okay, but I can understand from this number. I can understand if you give me number only, I can understand what does this enzyme do. Specifically, I will understand.
Yeah, understand. Thank you.
That is the impact. That is the significance of 4 number.
Thank you. Yeah.
Okay. Now see ligases. Now ligases, you can see ligases will join the bond. It will join between carbon-carbon bond. There will be join between carbon-oxygen. So this joining is called ligases. And coupled with always will be utilization of ATP. Hydrolysis of phosphate bond of ATP. So always utilize ATP. Remember, this ligase will always utilize ATP. So let's take the example, very good example. This is a very good example. This is one of the amino acyl tRNA synthetase. Amino acyl tRNA synthetase. There are 20 amino acyl tRNA synthesis you will study in translation in genetics. So if you understand this enzyme, it will help you understanding genetics also later. Translation. You can see this enzyme actually amino acid. This is one of the amino acid tRNA. So this is one amino acid. You can see. This is enzyme. This is one of the amino acid. You can see tyrosine is one amino acid. This tyrosine is being added to tRNA. So it will be joined by utilizing ATP because it is a ligase. So it will convert what will the result product will be lysyl, sorry, tyrosyl tRNA. So this is tRNA. This is tRNA. This is tRNA is clover leaf shaped. Now tRNA, then one amino acid will be chosen. So this will be the product by utilizing what? ATP. So this is ligase. It will join the bond. And here you can see ATP is being converted to AMP, 2-diphosphate, 2 high energy bond is being utilized. And this is a very specific enzyme. You will see about 20 types of amino acid tRNA. And this is rate limiting is one of the, this is the first step of the translation. This is called activation of, this is the step called activation of amino acid. So this is activated amino acid. I am giving you extra information so that it will make you easy to understand translation process. Did you understand? This is tyrosine tRNA synthetase. So you can see tyrosyl tRNA here. So this is ligase, one of the 20 bonds. So this is the enzyme commission number 6111. Now this is a very important for me. So you can see this is a pyruvate carboxylase. So pyruvate carboxylase, you can see this carbon dioxide is being added here, and this becomes this type of compound. This is pyruvate, 3 carbon compound, 1, 2, 3 carbon. This is pyruvate. So 1 carbon dioxide is being added here. This becomes pyruvate carboxylase. Now what comes here? Biotin will help this reaction, and ATP is there. So remember, biotin enzyme is coming for all carboxylases. There are 4 types of carboxylases only in our body. If you remember this reaction, it will be applied for all carboxylases. All carboxylases utilize biotin for carbon dioxide. You can see, if you see, there are 3 things, A, B, and C. So these carboxylases, if you remember mnemonics, A, B, C for this carboxylase, so you will remember the what function this carboxylase do. This is a ligase. It utilizes ATP and biotin to do carboxylation. It utilizes ATP and biotin to do carboxylation. So remember this carboxylase very well. Will you remember the function of this carboxylase? Tell me.
A, B, C.
Yes.
Using
Therefore,
ATP,
it utilizes ATP
TB,
and
then
biotin
and then we got
to do
the
carboxylation, to add carbon dioxide, to add carbon dioxide. It utilizes ATP, biotin,
And carbon dioxide.
to add carbon dioxide.
Where is biotin here in this?
So I have not written here, but you can write yourself. Okay.
Okay.
I have not written here. You can write yourself in note. So it is very important. Biotin is very important here. Got that? This reaction will be also repeating. That is why I have mentioned this reaction here. Got that? So I'll move to next slide.
Dr. Santosh, please.
Yes, tell me.
Carboxylase is in what group of enzymes?
So this is an enzyme, pyruvate carboxylase. This is the name of this enzyme is pyruvate carboxylase because it carboxylate. It adds CO2 to pyruvate group.
Yeah, what group will it be from 1 to 6?
This you can see here.
6.
Ligase, yes.
Okay.
Because it is utilizing ATP also. I told you A, B, C. It is utilizing ATP. And with ATP, it is also utilizing biotin. Okay. Ligase always utilize ATP. It is placed into group 6. Got that?
Doctor, I have a question. Do all the time ATP come like the opposite side, like the equation reverse?
Okay.
No.
Okay.
With
You are
ATP.
confused. You are confused with the reversible. Sorry. This is not reversible reaction. This is irreversible reaction.
Oh, okay. Yeah.
Okay. I'll change this. Thank you. Thank you for actually focusing me on this. This is
Yeah.
not reversible reaction. This is irreversible reaction. Okay. Got that? These are ligases are reversible reaction. Understood? Okay. So you can see the difference between lyase and ligase is utilization of ATP is very important. ATP utilization. Ligase always utilize ATP, but lyase never utilize it. So these are the, you can go through, these are very important enzymes given here from USMLE first aid book. So you can go through this book. So I have already mentioned many things. Phosphorylase, you can see dehydrogenase, hydrolase, hydroxylase, you can see hydroxylase, then uh, uh, many synthesis. You can go through this yourself. So kinases especially, I have a special focus on kinases and then phosphorylase. I've already explained. So go through this question. Which of the following group of enzymes always require coenzyme for their action? Always require coenzyme. A. Okay. So which of the following group of enzymes involved in production of ATP for survival?
Ligase.
D. It's
D,
D.
ligase.
Ligase will utilize, it will not produce. So oxidoreductase. Okay, because you know catabolism, oxidation, any oxidation will produce energy. Oxidation of fuel will produce energy. The same thing happens in our body. So oxidation reaction, when glucose converts into CO2 and H2O, what happens? Then energy will release, ATP will be produced. So what is happening basically? So oxidation is happening. Because of this oxidation is the main reaction that will release ATP. Okay, this principle, this concept we will use during our during our pathway study. So wherever you require ATP is produced because of this oxidation reduction reaction. We will talk about this in detail in our pathway. So which
So
of the
we expect
following?
this kind. We expect these kind of questions on the Monday test.
No, this type of question I will make you practice. So this type of question actually you will be asked a question on kinases. What is kinases? You will ask in question what is phosphorylase. You will be asked question on this concept that is I'm providing here. Okay, you will ask question which of the following. Yes, you can be asked this question also, but based on this concept I want to refresh your concept. So this is basic, very basic things, and actually USMLE never ask question directly from enzyme. USMLE connect this the concept of enzyme with pathway, then ask the question. Because you have not studied pathway, so I have to give my own question. Did you get that?
Yes.
Okay. So just I want you to go by very tiny things. Then after you understand things, then you will play a big game. Got that? This is the preparation I am doing to go for pathway metabolism. Which of the following group of enzymes are involved in digestion of food in the intestine?
Hydrolysis.
Yes, hydrolysis. So kinases fall in which of the following group?
Lysis.
Kinase. It actually transfer phosphate from
Transferase.
D.
Yes, transferase. Okay, kinase. All kinases are transferase. Remember this. So what is the role of enzyme named with kinase?
Transferring of
They are transferable to phosphate
Phosphate to
group to substrate.
substrate. They transfer phosphate from ATP to substrate. This is kinase. Okay. what is the role of enzyme named with phosphorylase?
Phosphoric acid.
Addition of phosphate.
Yes, they will add, actually, they will break the bond by using phosphate. Do you remember this? So this is phosphorylase. They will break the bond between two glucose molecule using, or two other molecule using phosphate. This is phosphorylase. So most of the carboxylases in the body are part of which of the following group? Carboxylases.
Ligase. Ligase.
Ligase. Very good. Ligase. So which of the following group of enzymes always utilize ATP? Always.
Ligase.
Yes. Ligase always utilize ATP. So carboxylases utilize which of the following vitamin as coenzyme?
Biotin.
Great.
Biotin.
Biotin. Yes, biotin. A, B, and C. Carboxylase utilize ATP, biotin to add carbon dioxide, to do carboxylation. A, B, C. If you remember A, B, C, you will remember carboxylase. So now let's talk about factors affecting enzymes a little bit. So there are 5 factors that affect enzyme. So among these factors, these 2 are very, very important. pH and temperature. pH and temperature are very important. So I have told you pH and temperature. So your body is, remember again, your body is actually integrated molecule performing in a very complex manner. So you are nothing but complex, actually accumulation of molecules. So you are enzymes. In that sense, you are also enzyme. So if enzyme function is very particular temperature and pressure, and that is why you cannot live in extreme temperature. Because enzyme cannot live in extreme temperature, cannot function in extreme temperature. That is why you cannot live in extreme temperature. So this is the significance of understanding factors affecting enzyme. So these are the factors affecting. If you increase the enzyme concentration, the rate of reaction will increase. If you increase the substrate concentration, to some extent, rate of reaction will increase. If you increase the coenzyme, so to some extent, the rate of reaction will increase. If you increase the temperature, so optimal temperature, the reaction rate will be highest. And then below and above at that particular temperature, the reaction rate will be slow. Same thing goes for pH. Let's talk about this. So you can see, let's give me only 1-2 minutes. Enzyme concentration, you can see if you increase enzyme concentration, then rate of reaction will increase. The more important is last 2. Temperature and pH is more important. And if you increase the substrate concentration, then the rate of reaction will increase up to certain degree, then it will be stagnant. I will talk about this in detail in next class of enzyme kinetics. So coenzyme concentration, if coenzyme is bound tightly, so it will act as enzyme. If coenzyme is bound loosely, so it will act as substrate. So reaction curve will be like this. This is hyperbolic curve. Remember the name of curve also. This is hyperbolic curve, this is a straight line curve. So in case of coenzyme, you will get both type of curve, hyperbolic and straight line. Temperature, main important thing is temperature. So you will see in temperature, you will see in bell-shaped curve. This is called optimal temperature. Below that temperature, the reaction rate will be slow. Above that temperature, reaction rate will be slow. So this is optimal temperature. Our optimal temperature is 30 degrees Celsius. The reaction rate is highest here. Now, there is something called rule of Q10. This is a very important. So with increase in temperature every 10 degree, by 10 degree, there will be doubling of reaction rate. Means the velocity is doubled when you increase the temperature. That is why if you see in summer season, food actually get damaged early because of bacterial enzyme activity, high activity. So with each rise in each 10 degrees Celsius, there will be doubling of reaction rate. And same you can see pH. For pH, you can see this is the optimum pH. Below which and below above that also there will be problem. Why? Because pH pH changes the protein structure, electrical charges on protein, because pH is proton, and that is why electrical charges of protein will be altered. That is why the enzyme activity will be very particular at particular temperature. It will decrease below a certain fixed temperature, pH, and also decreases above optimum pH. The rate of reaction decrease below optimum pH, also decrease if you increase pH. Same thing goes for temperature, because temperature also has the same effect on protein structure. So with this, did you all understand the factors affecting enzyme? Remember the curve, okay? Curve will be, so you can see 4 types of curve only. Only straight line curve, other is hyperbolic curve, and then bell-shaped curve. 3 types of curve only, you have to remember. If you remember curve, you'll remember factors affecting enzyme. And why pH and temperature, change in temperature, pressure, and pressure causes decreased enzyme activity. So you'll have to remember.